This section we're going to discuss lipids and some of the things that the categories and ways that we can make some of these lipids. So we're going to start with, we're not going to go into how we make waxes, but that is a type of lipid. We also have triacylglycerols, glycerophospholipids. So some of these things that are found in your cell membranes. We have sphingosine derived long backbone chain. Some of these are found in the membranes as well. We have steroids, and that can be used in membranes for cholesterol, kind of fluidity of membranes, and then also used as a chemical messenger. And echinocioids, another chemical messenger, or it also can be used for inflammation control, pain relief, that these are kind of used in that. And we'll talk about the synthesis of some of those kind of compounds as well briefly. I'm going to dig into some of the classical lipids that are more common here. Waxes, we have this functional group of the ester functional group here, where we just kind of have two long chains off of both sides of that. Triacylglycerol, these are things that a lot of times if you actually get your cholesterol checked, they're looking for triacylglycerols. These are components of fat that are found in our adipose tissue, our fat tissue. Again, we have the ester functional group, and then big long chains, right? You see CH2-14. So we have these big, huge, long, straight chains. They can be saturated, where there are no double bonds, unsaturated, where we have double bonds within them. This is an example of a triacylglycerol, or as opposed to a diacyl or a monoacylglycerol, where we have big chains of these off of all three of these components. Here's the backbone, is actually glycerol, like this, where it just has all of the hydroxide groups off of it. So this is glycerol, kind of the backbone for the triacylglycerol component, triacylglycerol. Then down here we have cholesterol, and we'll talk about the synthesis of that, and prostaglandins. So we're going to focus on more the cholesterol synthesis, but these are some other subgroups of those compounds. I also want to briefly just give you an overview of phospholipids and glycolipids, and these are some of the things that are found in our cell membranes. And there's a difference between, if you have a glycolipid, we kind of have these similar backbones, right? Glycerol, but then when we get to different types, we'll have sphingosine, which looks a little different, similar to glycerol, but a little bit different, where you have these fatty chains coming off of them. Glycolipids have sugars, glyco, right? The sugars attached to them covalently. Sphingomyelin has a phosphate and then an amino acid or amino alcohol on the top of it. And then glycerophospholipids have that glycerol backbone and then an amino group or alcohol, other different kind of polar. These are always polar head groups. Kind of have this alcohol group in the middle that is used to attach these acyl groups on the bottom end. And then below, just instead of just having these little cartoons or these little boxes, I actually show you what the options are for those green groups or those yellow groups or the blue groups. So you can see this sphingosine is a longer chain. It looks a little different, right? Instead of just alcohols, we have an amine group here. So this is actually an amide. It's an amide functional group here instead of a ester functional group. So a little bit different, but kind of similar idea. But instead of coming off of these alcohol groups and making esters, we're making amide functional groups down here with glycolipids and sphingomyelin. So we'd have to actually obviously have different enzymes that can cut amide bonds versus ester bonds if we're having enzymes cutting these versus these over here. So it allows a little different control and regulation of the metabolism of these different types of lipids. So where do all these fats come from? Where are they made? What happens here? Adipocytes or fat cells can't synthesize glycerol 3-phosphate from glycerol. So it can be synthesized from dihydroxyacetone phosphate or DHAP. So they can synthesize the triacylglycerides as long as there is available this DHAP. And this pathway is called glyceroneogenesis. And so that's where we kind of need to have this glycerol available to it here with the fatty acids to create that triacylglycerols that we just. But we're not going to focus on that pathway. We're going to mostly focus on the fatty acids and those big long chains of carbon and how they are made, and then we can kind of talk about how those longer chains are put together or break it down into energy. And basically the big picture ideas, we have triacylglycerides, glycerols, fatty acids, and acetyl-CoA is this linchpin for so many other processes in our body. Where are they made? So when we're building up, that's the synthesis. That is going to happen in the cytosol. So this is happening in our cells, cytosol. And so if this is our cell and our nucleus, the synthesis is mostly happening in this part. When we talk about, now this isn't to scale, obviously our mitochondria aren't as big as our nuclei, but the oxidation, the breakdown, this all happens inside the mitochondria. So these things are found, and then right away we reduce coenzymes, and we can get those in the electron transport chain to generate ATP. Versus if we're going to build stuff up create fatty acid tails, that's going to happen in the cytosol. And in terms of both of these oxidation and synthesis processes, acetyl-CoA is a central player in the metabolism of fat, and we'll talk about that. You'll see this a lot. We're actually going to start this whole process looking at the synthesis of fats rather than the breakdown of fats, because I think this makes sense, because we have already talked about in terms of once we get done with glycolysis, when we eat sugar, we make pyruvate, and then there are a series of steps that then allow us to generate the acetyl-CoA. So this is acetyl-S-CoA. This is what we kind of end up with before this goes into the citric acid cycle. So we can continue with this process in the citric acid cycle and use this for creating ATP. But if there's excess acetyl-S-CoA, then what do we do with it? Well, we can start making fat with it. So if we have too much of this, we start making fat. And the first step is the transfer of the acetyl group from acetyl-CoA to a different carrier protein, the S enzyme 1, and you can see that here. So we're going to do transferase reaction. We're swapping out this hydrogen for this acetyl group, and then the S enzyme 1 carrier protein is going to be attached to the acetyl group. So now we have acetyl ACP, or the acyl carrier protein, and then we're going to, so this is slightly different than S-CoA, but a similar kind of idea. It uses a sulfur, and there's a covalent bond that's being made. It's just a different coenzyme. So the next step is what we're going to do here is essentially add this bicarbonate group. We can see the carbon, and we have 3 oxygens. We're going to attach it to this back end here, and so that is going to give us this other group that you have this carboxylic acid functional group on the end. So once we have malonyl-CoA, then we do another swap here between this S-acyl carrier protein and malonyl-CoA, and we can then have malonyl acyl carrier protein is attached with this. We reconstitute that coenzyme. So again, we have excess of all of these acyl groups, because if we have a ton of energy, we're breaking down glucose, we're making a lot of acetyl groups in our body. So then we'll have a lot of these acyl ACP functional groups, and we'll have made a lot of this malonyl ACP. And this next step kind of seems weird, because we just put this carboxylic acid end on here, right? But we're actually going to now get rid of it. But that process is going to allow us to attach both of this kind of red part here with this new acyl group that we're going to attach. So this gives us some of the energy that we need to have this happen. So we're going to actually now release this. It was bicarbonate, but now it's carbon dioxide. We're going to release that CO2, and then we're going to attach this. And now what you've noticed is we're actually starting to build chains of carbon here. So we have a 4-carbon chain. So we're going in the reverse reaction. We're actually making these longer and longer now. Then this next step, we actually reduce. So we're going to reduce this carbon group. So it's going to go from a ketone to an alcohol. In that reduction here, we actually end up oxidizing an ADP. So we use up some energy, energy molecule here to then reduce this. And in the next step, we will then dehydrate. So we're making, this is technically a lyase reaction where we're removing a water molecule from this region to form this double bond between these two carbons. So we're dehydrating that bond. We're also, right, so then here we can then, we're going to, this is a fatty acid that is non-saturated. We're going to then add hydrogen here, so it's going to become reduced. We oxidize another NADPH molecule. So we're using energy here, right? Because this NADPH could have gone into the electron transport chain to generate that, that proton motive force that we talked about in ATP synthesis. Instead, we're using it to reduce this long chain. So we're getting rid of those oxygen bonds and we're making long chains here. So now you can see this 4-carbon chain, and it only just has that oxidized carbon at the very end that's attached to the acyl carrier protein. So we started with 2 carbons, one that was highly oxidized, ended up with 3 non-oxidized carbons, right? So we have fully reduced carbons here. So there's a lot of energy capacity in this, right? Because we used up this NADH in these different steps to actually get to this point. We also, earlier on, right up here, we used ATP to make this malonyl ACP. So we've used up a substantial amount of energy just to get a 4-carbon chain here. So energy was put into this chemical reaction. The result of this first cycle in fatty acid synthesis is the addition of 2 carbon atoms to the acyl group to give 4-carbon acyl group that's still attached to the carrier protein in the fatty acid synthase. So we have these 4 groups here, and this is just kind of a cartoon of that compound, and this is the biotin carrier protein, and it's going to kind of swap between, there's an arm here that what will kind of happen is it takes these acyl groups and it swaps it over here, and then it adds it, and it can swap back and forth as these things are attached and then transferred to another group, attached, transferred to another group. So this is a very efficient enzyme, and it's set up in a way where this kind of arm can spin around and have different regions on it. So it's there's multiple components of this protein enzyme that allow this all to happen very effectively and efficiently because it's all kind of in one spot. Kind of think of it as like a little factory. So you have one thing that happens, and then you attach it here, and then it kind of swings back, and you kind of reform that, and you can kind of keep going and going in a quick order. So now we're back at this 4-carbon group, this chain, and when we have more malonyl-CoA, then we can start adding to this chain pretty effectively. So we have this S enzyme complex, we have another SACP complex, and then we can kind of, you can see where these red groups, we have these red groups are being added on to this chain, and we can continue to grow this chain by 2 carbon atoms every time we release one of those CO2s, so this kind of end pops off like it did last time, and then we get the 2 carbons chain growing. We can repeat all those steps, and then we reduce, right? We reduce that carbon to the CH2 instead of the carbonyl group, and then we have a grown chain, and then we can repeat that until we get up to about 16 carbons. Palmitoyl group is what that's called when it gets up to 16. That's about as far as this enzyme can actually hold on to this carbon and grab onto these things, because then this isn't big enough to handle that long of a chain after it gets past 16. So that's about as long as we get for carbon chains in our bodies. This next step here, I just want to briefly talk about the synthesis of this glycerol. This can come from glycolysis, or I mentioned that other pathway that we can make glycerol. We're not going to focus on that, but I do want to talk about, once we have these big chains of fatty acid, right? So that's what we're doing here. This is a big fatty acid chain. It's attached to an S enzyme complex right now, but right, this is going to be that group that we're going to attach. That's going to be this group here. And remember, glycerol is normally that carbon with the OH group here, right? So essentially that's going to be the oxygen. This is the acyl group that we're going to attach. So we're going to talk about how we get this to work out now. So after we have, we're just going to assume that we have this glycerol 3-phosphate from glycolysis cycle. We're going to then start to add some of these chains. So this R group, right, that's a big long chain of carbon. we have this this carbonyl group that's attached to a sulfur that's in this coenzyme complex. We're going to transfer that acyl group onto this top chain of the glycerol. And so we're essentially going to have this group here, this oxygen, is going to be attracted to that carbonyl carbon, and it's going to be able to pop off this group from this sulfur here. So then we'll release that, and we transfer the hydrogen from the hydroxide group, comes over here, and then we transferred that acyl group onto the glycerol. So that's what we have here. Then we have this monoglycerol phosphate group. We then can do that with another chain and another chain, so we can do that 3 times, so we can get that triacylglycerol. This is a diacylglycerol right now, so that's the same process. We do a transferase reaction to then just transfer on those groups onto those different chains. So this phosphatidic acid, you notice this isn't in a hydroxide group, right? We have this phosphate, so we have to take that phosphate off. There's a transferase that releases that, generates a alcohol group here, and then we can use that final acyl transferase again. We can transfer that acyl group onto that oxygen right here. So then we end up with that triacylglycerol. So this last step obviously has to happen, so we have that alcohol group. So that same kind of enzyme can be used for all of these acyl transfer groups.
So we have looked at glycerol and fatty acids being combined to make the triacylglycerols. So that's what the process we just looked at. Now we're going to start looking at acetyl-CoA in a different pathway, where we look at not down to the citric acid cycle or over to ketone bodies or to protein, but to make steroids and cholesterol, which is a different part of the pathway, and it's done in a little different way here. We're going to talk about how we make these long rings. How do we make these big chains? Typically, they have that core structure of rings, and they look very similar to this compound or something like that. Cholesterol is an example of these kind of steroid, these hormones. Different hormones and vitamins are made from that. They can intercalate into the cell wall and change the kind of fluidity of that wall, and that's going to change some of those things. And then also some of the steroids and hormones. This is a cortisol, kind of a stress hormone. You can see that these can actually get through the wall and diffuse through the wall because they're fat, and they can get into here and get into the inner side of the wall and change the transcription of certain types of genes. So these are all kind of different things that these components can do. But how are they made? Where do they come from? To make cholesterol, we actually have to go down the mevalonate pathway, where we still start with acetyl-CoA. And I'm going to kind of go into this a little bit, but not too much. We're going to look more at where these go, and we're not going to walk through every single pathway. But essentially, you start with acetyl-CoA, and then we have the acetyl-CoA. We kind of combine that, and we create a bigger subunit of acetyl-CoA. So we take 2 of these, and then we combine them together, and then you get these kind of still oxidized subunits, but with longer chains of carbon. Then we add another one of these together and another water molecule, and we get this longer component, and then we continue to add these things together. Different animals and plants, and the Archaea 1 and Archaea 2, they can make some different derivatives and offshoots of this. But in humans, we kind of come to these 2 final products called isopentyl pyrophosphate and dimethylallyl pyrophosphate, or IPP and DMAPP. So the exact nature of this mechanism is not super important. We're going to come back and look at this a little more closely a little later. But for right now, just to kind of know, these acronyms just mean this long chain with 2 phosphate groups on the end. That chain right here, we'll kind of look at this a little bit. We see it's kind of got this a couple of carbons here, double bonds on them in different places. So we were talking about looking at plants. Terpenes are kind of they're part of the essential oils of different plants, and they're thought to potentially have some medicinal benefits. But again, these terpenes are kind of plant derived, looks very similar to these kind of derivatives of IPP and DMAPP. They're derived from plants. Terpenes can be kind of added subunits. You can kind of see the subunit is being repeated and repeated over and over again. So these are, there's 3 of those terpene subunits in here, and they have different names. And again, as I mentioned, often they are isolated from plant materials and parts of the essential oils. So just some kind of interesting side tidbit about terpenes and their potential role. We're not really sure what they all do yet or what their properties are, but it's kind of a emerging field in the sciences to try to figure out what these things might be doing in our bodies when we consume them. You'll notice that DMAPP and IPP, they look like terpenes, so they're kind of related biochemically. They're long chains with kind of a carbon off to one end and then double bonds around that. What we actually do is when we, during this initial state of this pathway, you actually start to glue these components together. So you see that red component and the blue component, the DMAPP and the IPP. We're gluing them together and making longer and longer chains. So as we make those long chains, you can kind of see this repetitive component as well. This is the coenzyme Q10, otherwise known as ubiquinol or ubiquinone. And so here's the oxidized form, which is called ubiquinone, the reduced form ubiquinol, ONE ending because it's a ketone, and OL ending because it's the alcohol functional group. So this is part of our electron transport chain. We talked about this as well when it grabs the electrons, right? It's going to be in the loss of electrons is LEO, so it's oxidized, and gain of electrons is reduced. So when it's gaining electrons in here, then it can transport it to the other components of the electron transport chain. That's its job in here. You can kind of imagine why it's fat soluble, because it's got this big long carbon tail where it's able to kind of go throughout this membrane here and transfer electrons. So it's a really important component of the electron transport chain. So back to the DMAPP and the IPP. So we can combine these to form the squalene, which is also going to be part of the component of this long chain here. That's part of CoQ10 that we can synthesize, but it is also then put together to form the precursor lanosterol and also the cholesterol component. So this kind of all comes together. I just did want to show you, this is the squalene, this is that long chain. You can kind of see when it's bent around like this, that's actually kind of what the precursor form is. But how do we make those chains? We actually have an enzyme here that will come in and oxidize this one subunit group here. We make a squalene epoxide. It's a very short-lived epoxide. So it's a 2 carbon and 1 oxygen grouping. So we have this triangle that's formed. We actually know that triangles are not very stable structures. So very shortly after this is created, we have electrons jumping over here. We have this turning into an alcohol. And we actually, because this leaves over here to form this bond between these carbons, then these electrons move here to replace those that were vacated to create this bond. And then we have this kind of reaction domino effect that then forms this whole, all of these rings within this squalene. So it's kind of an interesting mechanism for us to create that precursor of lanosterol. And then from that precursor, then we create cholesterol. From the cholesterols, we can make bile salts, cholesterol esters, testosterone, estradiol, so sex hormones, vitamin D3, lots of different things so that we can generate from this. So we can't make remember the A, D, E, and K vitamins, those are essential. Other plants and animals can, right? We can't make chlorophyll. So these are just all the possibilities of where this pathway can lead, depending on what type of organism you are. But these are kind of some of those things that from cholesterol, that's where we make our sex hormones as well. Terpenes I mentioned in that other slide as well. So this is where the cholesterol is coming from. Cholesterol in our body is carried through the bloodstream in lipoproteins, and that are two different kinds of lipoproteins: high density lipoproteins and low density lipoproteins. And if we look at these, LDL is usually called bad cholesterol because it leads to a buildup of cholesterol in the arteries. It's basically telling us that our liver has a ton of fat, and it's actually sending fat out of the liver in these groupings. It's made cholesterol, it's made fatty triacylglycerides, and it's releasing it into our blood to kind of get rid of it out of our liver because we have a buildup, sending it to adipose tissue and other tissues. HDL is kind of the opposite. It's called the good cholesterol. It carries cholesterol from other parts of your body to the liver so that the liver can remove that. And it's saying that there's not a lot of fat and cholesterol in your blood, but your liver is doing a good job trying to get it back into your system, into your liver, so it can convert that into energy or do other things with it. So usually having higher HDL levels are good, and lower LDL levels are good. When we have too much lipid, hyperlipidemia can cause disease. Again, we have kind of inflammation that can cause the plaques can build up and close up the arteries, which can lead to heart attacks and heart disease. And these are just examples of general. If you ever have your heart or your cholesterol checked, total cholesterol generally you want less than 200 milligrams per deciliter, LDL less than 100, HDL you want actually to be higher because that's the good type of cholesterol. Triacylglycerides under 150, and so again, it's right adults over the age of 20. So you guys in this class right now, it's about time to start getting your cholesterol levels checked. You can have high cholesterol even at the young age of 20. So here's an example of someone who had their cholesterol checked. So this is their cholesterol. So you should go back and review what I just said and decide if this person has good acceptable levels, borderline high, or high cholesterols based on these numbers. So this is one of the molecules, HMG-CoA, that we're going to look at, and this is one of the components of that mevalonate pathway. And what we do, they're called statin drugs. These drugs help patients reduce their cholesterol levels, and it does so by blocking the mevalonate pathway by kind of mimicking this kind of HMG-CoA compound. And this is actually where it is in this pathway we talked about that makes the IPP and DMAPP. So the HMG-CoA reductase is the target where that's actually inhibiting this enzyme that allows these metabolites from this process to that process down here. So one of the things to think about, if we're going to block this pathway, what else are we not able to synthesize? Are there any concerns with them? This is actually related to some of the side effects to this drug. Some of the negative impacts of taking statin drugs is kind of related to this question. So I'm not going to give you the answer right now, but it's something to think about in terms of what happens when we're taking statin drugs and the outcomes of that. And that concludes the first parts about lipids and the synthesis of different types of lipids. The next part is we're going to talk about the breakdowns of lipids and how we get energy from that. And essentially the big picture here is we're going from these triacylglycerides into fatty acids and acetyl-CoA. So how do we do that? And it's a little different than when we built it up in the previous few slides.